Okay, we are back again in a TYBSC Zoology Mammalian Histology class. We are continuously discussing about the kidney histology. In that, this is a part 3 lecture. In earlier lecture, we were up to the renal corpuscle. We discussed all these diagram here, glomerulus, juxtaglomerular complex, and Bowman capsule. Now we'll have a look at this certain textual point here about the glomerulus capsules. So visceral layer has podocyte that wraps projections around a single layer of endothelial cells of glomerular capillaries and form inner wall of capsules. Now this podocyte is one important point in a glomerulus. We'll discuss in detail later on in some after some slides. Then partial layer forms outer wall of capsule that we just discussed, that is the epithelial layer that form the capsule structures, and fluid filtered from glomerulus capillary enter capsular Bowman space. The in everything except RBC and WBC get ooze out from the capillary blood capillaries and enter into the capsule form, that is the Bowman's capsule space, and which then pass into the neck portion. Now here again we have some slide, and this slide particularly I want to show here because during the practical exam or during practical courses uh, you will observe this kidney section, and many times we focus such pointer on a glomerulus in a kidney and we ask you to identify the structure. So overall, by looking entirely on this structure, if you see this irregular vertical and horizontal curved portion with such crumble structures, you can identify this slide as a kidney section. Thereafter, once you identify this as a kidney structure, you will definitely come to know that this must be a glomerulus, and this way it will be easy to understand, to identify this slide as a kidney LS. Okay, this is again magnified. So all these are the convoluted tubules. These are the glomerulus. Okay. Now this is much more magnified renal corpuscles in a regular stained histological slide. So here we will get acquainted with the labels which we have seen in a diagrammatic drawing in earlier slide. So if we consider this as a Bowman capsule, this portion is a vascular pole. That means from where all the arterioles, afferent and efferent arteriole enter into the Bowman capsule. So this is a vascular pole and this is a urinary pole, because all that filtrate except RBC and WBC enter that liquid enter into the tubule system through this portion. So this is a urinary pole. Okay. Now this is a one artery. This is another artery here. In this afferent arteriole, you will find this specialized type of cell towards the glomerulus. So this is a macula densa. Okay. It's a part of a JG complex, juxtaglomerular complex. And all this structure is a capillary surrounded with the podocytes. A fenestrated capillary surrounded with the podocytes. And these round structure, if you carefully see here, these round structure, these are the RBCs. So if we try to see this same figure by eliminating this small rounded bits like structure, we will easily notice this dark nucleus here. This dark nucleus indicate the nucleus of podocytes over the glomerulus. Okay, here again is a renal corpuscle structure, and it shows the vascularization of the glomeruli, that is blood supply is shown over here. So this is only glomerular structure is colored here. That means only the bunch of that afferent efferent arterioles is colored here, and rest epithelial tissue are not that much colored So here you can distinctly separate these glomerulus with these other epithelial tissues. That means the tubule system. So we can say that this is now it's a glomerulus. So it's a renal corpuscle. Now again here renal corpuscle, but no distinct color stains are used in this slide. So here based on the size of epithelial tissue, we can understand that this is a collecting duct. Based on the epithelial tissue, we can see that this is a proximal tubule, while it is a large cuboidal epithelial cell with brush border are there. So it cover a lot of area in a cuboidal form, internal lumen area. So it's a proximal tubule. Now, as compared to this, if you see here, the epithelial tissue are much more subsile. No doubt they are cuboidal, but they don't have that brush border. So on that basis, we can say that this is a distal convoluted tubule, and this is a glomerulus, and here somewhere it is a that artery and macula densa and JG complex, everything happens here. So this is a vascular pole, and somewhere here there is a urinary pole will be there. This is again the urinary and vascular pole they are trying to show you in a glomerulus. So this is a neck portion of a Bowman capsule. So this is a urinary pole, and here somewhere there is a that vascular pole is there. That is because of the entry of arteriole and arterioles. So here again is a vascular pole, entry of arterioles, and you can see here very close to this arteriole there is a adjacent tubule is there, that may be a distal convoluted tubule come near to the vascular pole, that arteriole, to form the JG complex here. And can you notice where is a urinary pole that may be cut in a different plane, but that is opposite to the vascular pole here somewhere. Okay, here again in the renal corpuscle, light and electromicrograph image. So this is electromicrograph image, and this is a regular light microscopic image with a color stain with a eosine hematoxylin stain they have used here. Here it is a electromicrograph. Okay, so again this is a vascular pole. These are the clumps of that capillaries. These round structures are RBCs, and the dark violet blackish color are the nucleus of either endothelial cells or podocytes. And internally, this Bowman capsule is lined with the squamous epithelial tissues. So you will not see any such large bulging cuboidal cells here. It is all lined with the epithelial tissue. So from histological point of view, we must focus on the cell type here, how that cell type differentiates rather than the physiology of excretory or renal corpuscles. Now, if you see these renal corpuscles under electromicrograph, it look like this. These are all that podocytes covering these tubules. Okay. Now we were saying many times this podocyte thing, podocyte processes and peduncles, or podocyte in a glomerulus. What is that podocyte is? They are called podocyte because they have processed food-like processes. That each podocyte has few primary processes that give the cells a star-shaped appearance. These processes are wrapped around glomerulus capillaries and interdigitate with those of neighboring podocytes. That that means they forms like legs to attach with the another cells, and that legs are sort of a communication pathway between the cells. That legs when bifurcate, and that bifurcated parts then called as the pedicels. So here we have to little bit concentrate to understand what exactly is a podocyte. First, if you remember, just imagine that there is no podocyte and there is only a capillary present. So this pinkish color tube throughout the length, that pinkish color tube is a fenestrated endothelial capillary. So all these are the fenestrated endothelium of a blood capillary. This is the nucleus of that endothelial cells. So just imagine about this pink color structure, and over to this pink color structure there wraps this gray color structure. Now that will help you to imagine. So in cross section you will see like this, these are the endothelial cells, and there is a gap in between the outside these endothelial cells and between the podocytes. Now these podocytes are the another cell covering the entire capillary. Okay. So this podocyte is another cell type which engulf or cover the capillary from external outside. So this way they form a very close and increased contact surface with the oozing out blood from the penetrated endothelial capillary. Okay. Now, how these podocytes serve? The podocytes they form the processes, foods, that pedicles, foot-like processes which get sprayed. Now, here you can say they are sprayed over the surface of capillary, and that form the filtration slits with the diaphragms of opening and closing structure. So, how much amount of plasma should enter into the Bowman capsules are regulated by these podocytes. Okay, histologically, we don't have to go in a physiological functions, but the functions of podocyte cell is to control the amount of plasma entering into the Bowman capsules. That thing you should remember. And how it is a structure? It is a very proliferated growth, extensively forming processes and food structures, species structures, wrapping around the capillary present in a glomerulus. If you try to imagine, it will be very easy. See, this is pink color portions of capillary, and around these podocytes are wrapped. And one thing why they are called as a podocyte, because they form these all processes. Okay, now here it will be little bit clear. You can have an idea of that podocyte is. Okay, so here, if this is a podocyte, if these are the podocytes, a cell, they form such processes, and they cover this capillary. They wrap this capillary, and again they use all these processes which get sprayed over the capillary. Now this is a capillary running, and this capillary is wrapped with the podocyte processes, and they form connections between the opening pores of that capillary. They form junctions and gaps and a gate, so they can control the oozing of plasma through this capillary, because capillary as such they don't have any gate to close or open the uh, emptying of plasma into the Bowman capsule, but these podocytes have role to control the opening and closing of that gate to control the oozing out of plasma into the Bowman capsules. Now, again, these sections of a podocyte, this electromicrograph, these are the podocytes, and they form this. This is a glomerular capillary. So podocyte cover this capillary here. You can see this attachment line here. So this is attachment line between the podocytes and the capillary. You can see here. Here again in this area, this is a capillary. This is a fenestration or the opening between the capillary. This portion is all upper that endothelial portion. So this is from here from here to this side, everything is a blood capillary, and to this blood capillary, these podocytes processes pedicels are attached, anchored on the wall of external wall of endothelial cells, and that way they form these opening gaps, and this is the basement membrane over which these two things, from one side there is the endothelial cells fenestrations is there, and from another side there is the attachments of these pedicels forming the small pores that can be controlled. So the whatever the blood plasma ooze out through these penetrations, that don't directly get poured into the Bowman capsule, but it is controlled by the these podocytes diaphragms.